The primitive Earth, as it existed around 4.5 billion years ago, was a completely different planet from the one we know today. Instead of forests, blue oceans, and a breathable atmosphere, there was a geological inferno, a surface covered in molten rock, constantly erupting volcanoes, seas of incandescent lava, and a dense atmosphere composed of toxic gases such as methane, ammonia, carbon dioxide, and water vapor. There was no free oxygen in the air, nor was there an ozone layer to block the intense ultraviolet radiation coming directly from the young sun. Frequent impacts from asteroids and comets added to the catastrophic environment, further heating the unstable crust and releasing additional gases from the planet's interior. The recently formed Moon, the result of a violent collision between Earth and a Mars-sized body, still hovered closer to the planet, generating extreme tides that churned the first forming oceans. All this instability was not an obstacle to life, but rather a fertile ground for complex chemical reactions. Over the first 500 million years, Earth began to cool slowly. The first rains fell on the still hot crust, evaporating almost instantly, but eventually, after millions of years of gradual cooling, they accumulated in depressions, forming the primordial oceans. These liquid masses became immense natural chemical laboratories, where energy from lightning, volcanism, solar radiation, and geothermal heat acted on simple compounds dissolved in water, such as cyanide, formic acid, and metal sulfides. In this chaotic soup, organic molecules began to appear spontaneously, still far from life but representing the first building blocks of a long biochemical journey. The surface of the Earth remained unstable, but deep in these newly formed oceans, in hydrothermal vents, environments rich in minerals and energy emerged, perhaps more stable and protected from the extremes above. There, complex chemical reactions could occur with regularity and continuity, shaping increasingly organized structures. Beyond the planet's internal energy, the sun played an essential role in the process. Without the protection of the ozone layer, its ultraviolet radiation penetrated into shallow waters, provoking mutations and unexpected reactions among simple molecules. At the same time, the lack of free oxygen prevented the premature oxidation of fragile compounds, which paradoxically favored the formation of delicate organic structures such as amino acids and nucleotides. The extreme tides caused by the Moon's proximity created cycles of wetting and drying, especially in volcanic coastal regions where molecules concentrated, and polymerization reactions, the linking of small molecules into larger chains, were favored. These cycles may have been crucial for the emergence of the first life polymers, like rudimentary proteins and RNA fragments. Although still far from a living cell, the first ingredients were already gathered and chaotically mixed in a chemically boiling Earth. Comets and meteorites also played a crucial role in this equation. Rather than simply destroying the crust with their impacts, they brought with them complex organic molecules that had formed in interstellar ice or in chemical reactions within the celestial bodies themselves. Studies of meteorites found on Earth have revealed the presence of amino acids and hydrocarbons, building blocks of life coming from space. This supports the idea that the fundamental ingredients for life didn't originate solely on Earth, but may be common in the universe, scattered across celestial bodies traveling for millions of years before colliding with young planets like ours. The combination of what already existed here and what came from beyond may have been essential for the molecular diversity that made biogenesis possible. Rather than a single location, like a warm pond or a deep-sea vent, multiple environments may have acted simultaneously across different regions of the planet, developing distinct paths toward the organization of organic matter. Still, the mystery of the origin of life remains one of the greatest scientific enigmas. Primitive Earth offers clues, but no certainties. The oldest rocks we possess date back to about 4 billion years ago, and even in them, the evidence is fragmentary, based on carbon isotopes that may indicate biological activity, but could also have purely geological origins. What is known is that life somehow emerged in a hostile, chaotic, and brutal environment, where the laws of chemistry allowed the improbable, the transition from non-life to life. Even before the first cells, rudimentary structures were already capable of storing information, interacting with their surroundings, and even replicating imperfectly. The Earth, which seemed condemned to chaos, was in fact the warm womb of a biochemical miracle. And from this landscape of extremes, the journey of life began. Before diving into the theories about how life emerged on Earth, we must confront a fundamental question. What exactly is life? This seemingly simple question remains one of the greatest challenges in biology, philosophy, and even astrobiology. There is no universally accepted definition of life, but science operates with a set of criteria that help distinguish what is alive from what is not. Among the most agreed upon attributes are the ability to reproduce, to evolve through natural selection, to metabolize energy, to respond to the environment, to grow, and to maintain a highly complex and ordered internal organization. However, there are exceptions and gray areas in each of these criteria. Viruses, for example, do not have their own metabolism and cannot replicate outside host cells, yet they carry genetic information and evolve. Prions, infectious proteins, have neither DNA nor RNA, but they can propagate by altering other proteins. These blurred boundaries make the concept of life harder to define than it appears at first glance. From a molecular perspective, life is a self-organizing system that remains far from thermodynamic equilibrium. In other words, living organisms are not in a stable, motionless state. They are chemical machines that constantly consume energy and matter to maintain their internal organization, carry out functions, and resist decomposition. This organization is incredibly intricate. Living cells have compartments, membranes, molecular motors, signaling networks, and self-correcting mechanisms. More than that, all known life is based on four main building blocks: proteins, lipids, carbohydrates, and nucleic acids, and on chemistry dominated by the element carbon, whose ability to form long and complex chains is unparalleled. This universal biochemical architecture is one of the strongest clues that all terrestrial life shares a common origin, even though it has diversified in spectacular ways over billions of years. Another essential point in defining life is the presence of an inheritable information system. Every living cell carries within it a set of instructions that determine its structure, functions, and capacity to produce offspring. This information is encoded in molecules of DNA or RNA, which function as a highly precise chemical language. That language must be copied with reasonable fidelity, but also with room for occasional errors, mutations, which are the fuel of evolution. Reproduction with variation is what allows living populations to change over time, to adapt, to diversify, and to colonize new environments. Thus, one of the keys to life is not just to exist, but to perpetuate itself, to change, and to respond to environmental challenges. Life is ultimately a dynamic process that generates diversity from highly organized and replicable structures. Yet there is a deeper dimension. Life is not merely a collection of properties or molecules, it is an emergent process. Just as a symphony cannot be explained solely by analyzing individual musical notes, life cannot be fully understood by merely examining atoms and bonds. The interaction between simple parts gives rise to complex and often unpredictable behaviors. When the right molecules are arranged in the right way, something new emerges, something that metabolizes, that learns, that feels, that adapts. Life is a property of the whole, not just the sum of its parts. This idea of emergence is essential for understanding why the origin of life was not necessarily a magical or singular event, but a gradual process, where complexity and functionality increased in small steps until at some point the boundary between the non-living and the living was crossed. Therefore, when we study how life began, we are not searching for a magic formula, but trying to understand how sets of molecules could, through the laws of physics and chemistry, become self-sustaining, reproductive, and evolutionary systems. We are trying to identify the moment when a set of reactions became a closed system that defended itself against entropy, stored chemical memory, and reproduced with variation. We are seeking the first spark of biological continuity in a world governed by blind physical processes. The challenge is monumental because we cannot go back in time or directly observe the original event. All we can do is reconstruct, molecule by molecule, step by step, the transition from an abiotic world to a biological one. Understanding what life is the first step to understanding how it began, and perhaps one day how it might arise on other planets. The so-called primordial soup hypothesis is one of the most influential and enduring ideas about the origin of life on Earth. Originally proposed in the early 20th century by scientists such as Alexander Oparin and J.B.S. Haldane, it suggests that life began from a rich mixture of organic compounds dissolved in the shallow, warm and chemically active oceans of the early Earth. This soup, heated by geothermal activity and energized by electric discharges, ultraviolet radiation and solar heat, may have allowed the spontaneous emergence of complex molecules from simple components such as methane, ammonia, hydrogen and water vapor. Oparin and Haldane theorized that on a planet still lacking free atmospheric oxygen, a highly reactive element that degrades organic molecules, these substances could have accumulated in high concentrations over millions of years, creating the ideal broth for life's chemistry to emerge. The strength of the primordial soup hypothesis lies in its conceptual simplicity and its compatibility with the principles of organic chemistry. The absence of free oxygen in the ancient atmosphere and the abundance of energy sources would have created a favorable setting for chemical reactions that form amino acids, nitrogenous bases, sugars, and lipids, the fundamental building blocks of life. The primordial soup was not a uniform and homogeneous substance, but rather a dynamic and heterogeneous landscape, with concentrations varying due to tides, rains, droughts, and thermal cycles. In pools that dried and refilled cyclically, organic molecules could have become concentrated enough to react with one another, forming increasingly larger chains. Under such conditions, simple compounds could have transformed into rudimentary polymers, such as small proteins or RNA fragments, foreshadowing the emergence of the first biologically functional structures. The metaphor of the soup also helps illustrate the complexity of the original scenario. Instead of a single, straight and inevitable evolutionary line, there was chemical chaos, where millions of combinations occurred simultaneously, but only a few led to more stable, complex or replicable structures. In this pre-biological context, natural selection did not act on organisms but on molecules. Those that degraded quickly were eliminated, while those that persisted, accumulated, or replicated could give rise to new levels of complexity. The primordial soup was therefore a colossal and spontaneous laboratory spread across the planet's surface and driven by natural cycles, where molecules tested combinations blindly, yet governed by the laws of physics and chemistry. Over time, one of these combinations may have crossed the threshold between inert chemistry and biological activity. For decades, this hypothesis remained unverified, until, in 1953, scientists Stanley Miller and Miller-Urey conducted an experiment that provided direct support for the idea. They recreated an artificial atmosphere in the lab containing the gases believed to be abundant on early Earth, methane, ammonia, hydrogen, and water vapor, and applied electric discharges simulating lightning. After a week, the mixture revealed the presence of various amino acids, the basic units of proteins. The impact was immediate and historic. For the first time, it was proven that essential compounds of life could form spontaneously under plausibly prebiotic conditions. The Miller-Urey experiment became a cornerstone in the study of life's chemical origins and one of the strongest demonstrations that life could emerge naturally without supernatural or unknown intervention. Nevertheless, the primordial soup hypothesis also faces challenges. More recent studies suggest that early Earth's atmosphere may not have been as reducing as Miller and Urey assumed. Instead of abundant methane and ammonia, it may have been dominated by carbon dioxide and nitrogen, gases less reactive for forming organic compounds. Moreover, the dilution of the oceans may have been a barrier to the concentration of molecules needed to form larger structures. For this reason, many modern variations of the theory propose that specific environments, such as shallow volcanic lakes, hydrothermal vents, or areas with evaporation cycles, would have been more effective zones for molecular concentration and polymerization. Even so, the idea of a soup rich in life's precursors remains a powerful model because it captures the core concept. Life did not appear ready-made, but as the final product of a long process of chemical self-organization in a fertile and chaotic environment. The Miller-Urey experiment, conducted in 1953, is one of the most iconic experiments in the history of science. frequently cited as the first successful test of a hypothesis regarding the chemical origin of life. Stanley Miller, a young doctoral student at the University of Chicago, under the supervision of Miller-Urey, constructed an ingenious apparatus designed to simulate the estimated conditions of Earth's primitive atmosphere. In a sealed glass system, he introduced methane, CH4, ammonia, NH3, hydrogen, H2, and water vapor, the gases believed at the time to represent the early atmosphere. He then applied electric discharges to simulate lightning, one of the most abundant energy sources on early Earth. After just 1 week, the water inside the apparatus had turned cloudy, and upon analysis, it revealed something extraordinary: the presence of amino acids, including glycine, alanine, and aspartic acid, fundamental building blocks of proteins. The impact of the experiment was immediate and profound. For the first time, there was direct evidence that complex organic molecules essential to life could form spontaneously from simple compounds under conditions that might have existed on the primitive Earth. Miller's apparatus, despite its simple appearance, became a symbol of the experimental approach to the origin of life problem, a field previously dominated by philosophical and theological speculation. From that point on, the hypothesis of life's abiotic origin ceased to be an abstract theory and gained empirical support, fueling a new research area known as prebiotic chemistry. The idea that life could emerge through natural laws without the need for external or supernatural intervention found in the Miller-Urey experiment a scientifically credible anchor that endures to this day. However, as geology and paleoclimatology advanced, questions arose about the atmospheric assumptions behind the experiment. Modern studies suggest that Earth's early atmosphere may have been less reducing, meaning less rich in electron-donating gases like hydrogen and methane, and more neutral, composed predominantly of carbon dioxide, nitrogen, and water vapor. This shift in the atmospheric model implies that the original conditions simulated by Miller may not have been entirely realistic. Even so, the relevance of the experiment remains intact. Subsequent research has shown that even in neutral atmospheres, additional energy sources such as ultraviolet radiation, meteorite impacts, or hydrothermal activity could compensate for the lower reactivity and still allow the synthesis of organic compounds. Moreover, variations of Miller's original setup using different gas mixtures have consistently produced amino acids and other life precursors. A key aspect of the experiment lies in its demonstration that simple systems can spontaneously generate complexity. The fact that amino acids appeared in such a short time, with such few ingredients, and in a controlled environment, indicates that life's chemistry may not be an extraordinary event, but rather a natural possibility whenever the right conditions are met. This insight has led many scientists to believe that the emergence of life could be a relatively common process in the universe, provided that the essential elements, carbon, hydrogen, nitrogen, oxygen, and phosphorus, are present in an energetically active environment. The simplicity of Miller's experiment contrasts with the complexity of living organisms, yet it establishes a crucial link. It shows that there is no unbridgeable gap between the abiotic world and the biological one. The bridge begins with molecules like the ones Miller produced in a glass flask. Curiously, sealed samples from the original 1953 experiment were rediscovered in 2007. Using modern techniques such as chromatography and mass spectrometry, researchers reanalyzed the compounds and discovered that Miller had actually produced not only simple amino acids, but also more complex compounds that went undetected at the time due to technological limitations. This further reinforced the idea that a primitive chemical environment could generate a wide range of prebiotic molecules. Additionally, later adaptations of the experiment were tailored to simulate conditions found in hydrothermal vents, impact craters, and environments with drying cycles, all relevant to modern hypotheses about the origin of life. Even after more than 70 years, the legacy of Miller-Urey remains a foundational cornerstone in the science of origins. More than just an experimental milestone, the Miller-Urey experiment represents an epistemological turning point. It proved that the origin of life can be investigated with empirical tools, controlled tests, and reproducible data. It launched a tradition of research grounded in the idea that even the most mysterious phenomena of existence can be explored through science. Today, dozens of laboratories around the world continue exploring the paths that Miller opened, attempting to synthesize nucleotides, construct protocells, study molecular stability in extreme environments, and even simulate extraterrestrial oceans such as those of Titan or Enceladus. All of this began with a small glass chamber, a few sparks, and a bold question. Science, rather than giving us instant answers, allows us to build bridges, and Miller's experiment was one of the first solid steps toward understanding how life began on this small blue planet. The origin of life, in any plausible scenario, depends on the formation and accumulation of certain fundamental components, the so-called building blocks of life: amino acids, lipids, sugars, and nucleotides. These molecules, though individually simple in structure, are key pieces for constructing more complex entities, such as proteins, cell membranes, nucleic acids, and metabolic structures. Understanding how these blocks could have emerged spontaneously in an abiotic environment is one of the central goals of prebiotic chemistry. And surprisingly, over the past 70 years, scientific advances have shown that the natural synthesis of these compounds is not only possible, but can occur in a wide variety of environments, with multiple energy sources and differing chemical conditions. The complexity of life begins to emerge precisely when these molecules interact in increasingly organized systems, initiating a subtle but profound transition from pure chemistry to biology. Amino acids, for example, are essential for the construction of proteins, which function as enzymes, cellular structures, and regulators of vital processes. The formation of amino acids from simple gases, as demonstrated by Miller and many others since, is a robust process. In reducing atmospheres with methane and ammonia, amino acids form readily. Even in less reactive environments, sources such as ultraviolet radiation, cometary impacts, and reactions on metal-rich minerals can generate these compounds. Experiments in hydrothermal vents have shown that amino acids can also form in the absence of light, using heat and mineral catalysts like pyrite or iron oxides. Carbon-rich meteorites, such as the famous Murchison meteorite, also carry an impressive variety of amino acids, including some not used by terrestrial life, suggesting that life's building blocks are produced naturally even beyond Earth. Lipids, in turn, are molecules with amphipathic properties, meaning they have one part that attracts water and another that repels it. This unique characteristic allows them to spontaneously form micelles or lipid bilayers, structures very similar to the membranes of modern cells. Simple lipids, such as short-chain fatty acids, can be synthesized under prebiotic conditions and have already demonstrated in laboratory settings the ability to form bubbles that encapsulate other molecules. These lipid bubbles, known as protocells, are considered a crucial step in the compartmentalization of life's chemistry, that is, the separation between inside and outside, allowing chemical reactions to occur in protected and controlled environments. Additionally, lipids can self-organize in response to physical stimuli such as temperature and pH, providing a primitive foundation for cellular behavior without the need for genes or proteins. Sugars, especially ribose, which forms the backbone of RNA, present a more complex challenge for prebiotic synthesis. Nevertheless, recent advances have shown that even in the absence of enzymes, certain reactions known as formose reactions can produce sugars from formaldehyde in the presence of mineral catalysts like calcium or boron hydroxides. Although the yields of these reactions are low and the products unstable, specific environments, such as salt-rich lakes or areas with drying cycles, may have allowed for accumulation and natural selection of more stable compounds. Alternatively, some scientists suggest that sugars could have been delivered to Earth by meteorites or comets, where they formed through interstellar processes in cosmic ice and dust. The presence of complex sugars and alcohols in carbonaceous meteorites strengthens this hypothesis and broadens the range of possibilities for the origin of biochemical building blocks. Beyond these three main groups, there are the nucleotides, the basic components of DNA and RNA. They consist of a nitrogenous base, a sugar, such as ribose, and a phosphate group. The abiotic synthesis of complete nucleotides is particularly difficult, but research by scientists like John Sutherland has demonstrated plausible pathways for assembling them from simple compounds like cyanamide and formaldehyde. Sequential reactions catalyzed by minerals or facilitated by thermal cycles can produce functional ribonucleotides. This is highly relevant to the RNA world hypothesis, which proposes that RNA was the first molecule to store genetic information and also function as a catalyst. The ability of nucleotides to link together, form strands, and eventually replicate with some fidelity represents a turning point between ordinary chemistry and a truly evolutionary system. Taken together, amino acids, lipids, sugars, and nucleotides form the foundation upon which all known life is built. Modern science demonstrates that none of these blocks require life to exist. They can be formed in purely chemical, spontaneous reactions driven by heat, electricity, radiation, or mineral catalysis. The great mystery is not only how these molecules formed, but how they organized, interacted, and evolved together. Life is more than the sum of its parts, but those parts must exist first. The emergence and accumulation of life's building blocks represent, therefore, the initial and inevitable step in any journey toward biogenesis. Nature has shown on a cosmic scale that life's chemistry is everywhere, but transforming it into life, that remains the next mystery. The RNA world hypothesis is one of the most influential and debated ideas in modern biology regarding the origin of life. Initially proposed in the 1960s, and strengthened by experimental evidence in the following decades, this hypothesis suggests that before the emergence of DNA and proteins, RNA played a dual and essential role, storing genetic information and acting as a chemical catalyst. Unlike DNA, which serves only as a static data archive, and proteins which perform biochemical reactions, RNA is versatile enough to carry out both functions. This versatility positions RNA as an ideal candidate for Earth's first self-replicating genetic system, functioning as a bridge between pure chemistry and functional biology. In other words, RNA may have been the first truly living molecule, capable of copying itself, interacting with the environment, and evolving through natural selection. The foundation of this hypothesis rests on the discovery of ribozymes, RNA fragments with catalytic activity. Contrary to prior beliefs, it's not just proteins that can catalyze chemical reactions. RNA can do it too. In 1982, Thomas Tech and Sidney Altman demonstrated that certain types of RNA are capable of cutting, pairing, and recombining other RNA molecules without the help of enzymes. This was revolutionary. It proved that RNA, in addition to storing information, could also perform complex biochemical functions. This opened the theoretical possibility of a primitive system in which RNA molecules could replicate and evolve independently, without proteins or DNA. This initial system, still rudimentary, may have been the precursor to modern cells, a kind of molecular ecosystem where different RNAs competed for resources, interacted, and improved through random mutations and chemical selection. But how could RNA have arisen in an abiotic environment? This remains one of the most challenging questions in prebiotic chemistry. RNA consists of three parts: a nitrogenous base, such as adenine or cytosine, a sugar, ribose, and a phosphate group. The spontaneous formation of these parts is already difficult, but assembling them correctly into a functional nucleotide is even more complex. However, recent research has demonstrated viable pathways for this synthesis. In 2009, John Sutherland's group successfully demonstrated a plausible chemical route for forming ribonucleotides from simple compounds like cyanamide, glycolaldehyde, and phosphate, in reactions that occur step by step under geologically realistic conditions. This discovery not only restored the plausibility of the RNA world, but also showed that these molecules could have been assembled in volcanic lakes, thermal springs, or on catalytic mineral surfaces of early Earth. Another important aspect of the RNA world hypothesis is its potential for self-replication. Laboratory studies have shown that certain RNA fragments can catalyze the formation of partial copies of themselves, especially when anchored to mineral surfaces that help align the molecules. Even if these copies are imperfect, the process constitutes a system where variation and reproduction occur, allowing for natural selection. In environments with wet-dry cycles, such as intermittent volcanic regions, these fragments could concentrate, reorganize, and evolve into increasingly efficient forms. Once this dynamic was established, RNA may have formed cooperative networks of molecules where different ribozymes performed complementary tasks. Some replicated, others cut, others joined fragments. This modular and self-organizing system would have formed the foundation of the first molecular ecosystems that eventually evolved into primitive cells. Still, the RNA world hypothesis does not solve all the puzzles. RNA is chemically unstable, especially in aqueous environments and under high temperatures. It degrades easily, making its accumulation and long-term preservation a challenge. Moreover, efficient replication without enzymes has not yet been fully achieved in the lab, leaving a gap between what is theoretically possible and what has been demonstrated experimentally. For this reason, some researchers suggest that simpler precursor molecules, such as ANA, TNA, and PNA, artificial analogues of RNA, may have played the initial role, with RNA emerging later as an evolutionary refinement. Others propose hybrid systems, in which lipids, peptides, and small RNAs interacted from the start, each with a specific function, creating an emergent system in which life could flourish. Despite these challenges, the RNA world hypothesis remains one of the strongest candidates for explaining how life may have begun. It proposes a natural trajectory based on known molecules with replicable properties subject to selection. Even if RNA was not the very first living molecule, it may have been the first to integrate the two essential functions of life, heredity and catalysis. By placing RNA at the center of the origin of biology, this hypothesis brings the chemistry of life's beginnings closer to modern biochemistry. After all, even in contemporary cells, RNA continues to play central roles in protein translation, gene regulation, splicing, and even in essential structures like ribosomes. Perhaps in studying RNA, we are looking directly into a mirror of the past, into the first molecular voices that echoed through the silence of a lifeless Earth. The discovery of ribozymes, RNA fragments with catalytic capabilities, profoundly transformed our understanding of how life might have emerged from simple chemical systems. For a long time, it was believed that only proteins, especially enzymes, could catalyze the essential chemical reactions of metabolism. But in the 1980s, two scientists, Thomas Seck and Sidney Altman, demonstrated that RNA can also function as a catalyst. They identified RNA molecules capable of cutting, splicing, and reorganizing other RNA segments, all without the presence of proteins. This discovery earned them the Nobel Prize in Chemistry and paved the way for the hypothesis that, in a prebiotic world, RNA might have served not only as an informational molecule, but also as an active agent in regulating and transforming its surrounding chemical environment. Ribozymes thus became fundamental elements in reconstructing the possible early steps of life. Modern ribozymes are still found in living organisms and perform critical functions. One of the most important examples is the ribosome, the structure responsible for synthesizing proteins in all living cells. At the core of the ribosome, the reaction that forms peptide bonds, which link amino acids, is catalyzed by ribosomal RNA, not proteins. This means that even today, the heart of life's genetic machinery is powered by RNA's catalytic activity, a direct inheritance from a molecular past in which RNA was dominant. Ribozymes also appear in processes such as pre-mRNA splicing in eukaryotes, translation regulation, and even in the replication of certain viruses. This evolutionary persistence reinforces the idea that functional RNA is not an exception, but a deeply rooted feature of life's biological architecture. In laboratory contexts, scientists have succeeded in creating synthetic ribozymes that replicate short RNA sequences, join nucleotides, and even perform autocatalysis, that is, catalyze the synthesis of copies of themselves. Although these systems are still limited in efficiency and length, they demonstrate that rudimentary self-replicating circuits are chemically feasible. When small variations are introduced in these experiments, it is observed that some ribozymes replicate faster or with greater fidelity than others, which sets the stage for a primitive form of molecular natural selection. Different variants compete for substrates, persist for varying durations, and eventually dominate the population. From this, a dynamic scenario emerges in which molecules not only exist, but evolve. This kind of experimentation is foundational for understanding how living systems could emerge spontaneously in abiotic environments. One of the most fascinating contributions of ribozymes to the theory of life's origin is the concept of autocatalytic circuits. Imagine a set of different RNAs, each catalyzing the formation of the next in a closed loop. A catalyzes B, B catalyzes C, and C catalyzes A again. This type of molecular network not only enables reaction continuity, but also facilitates the amplification of advantageous variations, essentially a primitive form of evolutionary memory. Such systems could become encapsulated within lipid membranes, forming protocells that carry simple metabolic networks powered by ribozyme catalysis. These protocells could grow, divide, and compete for resources, gradually approaching the threshold of functional life. In this model, life did not begin with a perfectly formed cell, but with self-replicating molecular networks that acquired complexity step by step. Despite experimental progress, the path to a fully self-replicating ribozyme has not yet been completed. The molecules produced so far are short and have limited efficiency, mainly because RNA is chemically unstable and prone to breaking down in wet environments or in the presence of metal ions. Moreover, the ideal conditions for replication do not always align with those that favor catalysis, raising questions about how both processes could have coexisted in the prebiotic world. Still, progress continues. New approaches include using mineral surfaces as replication scaffolds, thermal cycling to favor specific reaction steps, and microfluidic systems that simulate natural environments such as volcanic fissures or bubbles in thermal springs. Each experiment brings us one step closer to understanding how an ancestral RNA could have bridged the gap between early Earth chemistry and the beginnings of biology. Therefore, ribozymes are far more than just a biochemical curiosity. They are functional proof that life could have been built upon RNA alone, at least in its earliest stages. They show that nature did not need to wait for complex proteins to take the first steps toward biological organization. RNA on its own can carry information, catalyze reactions, fold into three-dimensional structures, and even replicate. Even though modern life has evolved into a more efficient system with specialized DNA and proteins, the molecular memory of functional RNA still lives on in every cell. At the beginning of everything, long before there were eyes to see or brains to think, ribozymes may have already been there, silent, replicating, catalyzing, testing pathways that would eventually lead to the life we know today. If RNA offered primitive Earth the ability to carry information and catalyze reactions, proteins, composed of amino acid chains, brought a new level of functional and structural versatility to the chemistry of life. The emergence of proteins marks a key moment in the transition from prebiotic chemistry to complex living systems, as proteins carry out virtually all essential tasks in modern cells. They function as enzymes, transporters, molecular motors, sensors, and even structural elements. However, understanding how the first peptides, short chains of amino acids, arose before the existence of the genetic code and the cellular translation machinery remains one of the great enigmas of biogenesis. The formation of functional proteins with specific amino acid sequences today requires a highly coordinated system involving ribosomes, messenger RNA, transfer RNA, and dozens of enzymes. But in a world without cells, how could such complexity have begun? The most promising answer comes from experiments showing that amino acids can spontaneously link into short chains, especially in environments with cycles of drying and heating, such as shallow volcanic pools or hot mineral surfaces. In these places, the concentration of amino acids increases as water evaporates, allowing thermodynamic forces to bind the molecules together, forming peptide bonds. Minerals like clays and metal oxides act as natural catalysts, accelerating the process and protecting the molecules from degradation. In the lab, researchers have observed the formation of chains containing 10 or even 20 amino acids, using only heat, pressure, and simple compounds. A plausible prebiotic synthesis that supports the idea that the first peptides did not arise inside a cell, but outside, as rudimentary structures that spontaneously acquired useful functions. One of the most intriguing roles attributed to early peptides is that of molecular stabilizers within catalytic RNA networks. Since RNA is a fragile molecule, prone to breaking down easily, certain peptides may have acted as supports, scaffolds, or even shields, increasing the stability and functionality of ribozyme-based systems. This symbiotic relationship between RNA and peptides may have marked the beginning of molecular cooperation, with peptides enhancing catalysis and RNA guiding the assembly of new peptides, even in a primitive way. Studies show that simple peptides can already accelerate specific chemical reactions, even without a perfectly defined sequence. This suggests that catalytic functionality came before genetic precision. In other words, the first peptides didn't need to be extremely specific to be useful. They just needed to be useful enough to persist, grow, and be copied in some form. Another possible path for the emergence of functional peptides involves primitive chemical codes, based on direct interactions between RNA and amino acids, without the need for ribosomes. Some RNA sequences have shown, in laboratory conditions, a spontaneous and repeatable affinity for specific amino acids, forming natural RNA amino acid pairs. These pairs could represent the embryo of what would later evolve into the universal genetic code. In a prebiotic scenario, RNA fragments and amino acid chains might have interacted on mineral surfaces or within lipid compartments, creating RNA peptide complexes that replicated and evolved together. Molecular natural selection would then favor the most stable, efficient, and self-perpetuating complexes. Thus, life would not have needed to begin with fully complex and precise proteins. Simple, versatile peptides could have participated in early life chemistry and progressively evolved into more refined forms. Furthermore, the structural versatility of proteins may have given protocellular systems a new functional dimension. Peptides with lipid affinity could have aided in the formation and stabilization of primitive membranes. Others with catalytic properties might have accelerated RNA replication or facilitated the conversion of basic compounds into more useful molecules. In dynamic environments, where heating and cooling cycles alternated between wet and dry phases, peptides might have also acted as molecular memories, preserving useful structural patterns across cycles. Gradually, chemical selection and self-organization would have favored systems capable of integrating RNA, peptides, and lipids into a cohesive and functional unit. The emergence of stable and adaptive proteins would have opened the path to modern biology, now dominated by a nearly infinite variety of enzymes, hormones, antibodies, and ion channels, all descendants of those primordial peptides. Ultimately, the emergence of proteins was both a consequence and an accelerator of nascent life. They enabled faster, more diverse, and more responsive chemistry. But for this to happen, all it took was for some amino acids to link together in warm, moist, and energy-rich environments, creating the first chains that, unknowingly, would give rise to everything. Today, every living cell carries thousands of specialized proteins, each precisely encoded in its genes. But behind that complexity lies the molecular memory of a time when simple amino acid chains began to dance around RNA, taking the first steps of a choreography that would transform the planet. The slow chemistry of the past gained arms, legs, and momentum, and when peptides joined the molecular dance, life made an evolutionary leap, discrete, silent, but irreversible. The formation of the first cellular membranes represents one of the most crucial milestones in the transition from prebiotic chemistry to organized biological systems. Without a clear boundary between inside and outside, any attempt to sustain stable metabolism, coordinated replication, or chemical inheritance becomes practically impossible. That's why the emergence of structures capable of compartmentalizing chemical reactions and protecting molecular assemblies from the external environment was essential for the rise of protocells, rudimentary systems that preceded modern cells. Interestingly, basic membranes do not require genetic or enzymatic machinery to form. Simple lipid molecules, especially fatty acids, possess a fundamental chemical property that enables spontaneous self-organization into spherical structures when placed in water. This property, known as amphipathy, results in bilayer membranes that fold in on themselves, forming small water-filled bubbles, the lipid vesicles. These vesicles, often referred to as protocells, have strikingly similar features to modern cell membranes. They define an internal space, are semi-permeable, can grow by absorbing new lipids, and can even divide spontaneously under certain conditions. In the lab, scientists have produced vesicles from simple fatty acids obtained via abiotic synthesis or extracted from meteorites, reinforcing the idea that the basic ingredients for membranes were available on the early Earth and even beyond. When these protocells encapsulate RNA molecules or other reactive substances, they create genuine microreaction environments where molecular interactions can occur more efficiently and with greater stability. In natural settings like hot springs, volcanic shores, or intermittent pools, such structures could have emerged, expanded, ruptured, and reorganized millions of times, functioning as natural incubators for the development of nascent life. The great advantage of membranes is their ability to concentrate molecules and promote selective isolation. In a primordial ocean filled with organic compounds, molecular dispersion would have hindered accumulation and continuity of reactions. Inside a vesicle, however, interacting molecules are kept close together, increasing the chances of sequential and complex reactions. Furthermore, the internal environment can differ from the external one in terms of pH, salinity, and reagent concentration, creating chemical gradients that are fundamental to metabolism. Some protocells also show the ability to absorb nutrients, concentrate energy compounds, and even respond to environmental stimuli, such as changing their permeability in response to heat. These emerging properties, though rudimentary, mark the first steps toward functional compartmentalization, a principle that remains central in modern biology, from simple bacterial cells to complex multicellular organisms. Another fascinating aspect of primitive membranes is their evolutionary plasticity. While today's cells have highly sophisticated membranes with transport proteins, receptors, and specialized channels, protocells were extremely simple. Even so, these structures could grow by absorbing lipids from the environment or from other vesicles they encountered. Some even divided naturally when reaching a certain size or under agitation cycles. Random but continuous division, combined with some form of rudimentary internal molecular replication, such as self-replicating RNA or catalytic peptide networks, could give rise to molecular lineages with small internal variations passed down to descendants. This opens the possibility for encapsulated molecular Darwinian evolution, meaning natural selection, acting not just on isolated molecules, but on semi-stable functional units with internal metabolism, replication, and defined boundaries. Membranes could also have favored cooperation between different types of molecules. RNA fragments that catalyzed different reactions, peptides that stabilized internal structures, or lipids with varied physical-chemical properties could coexist within the same protocell. This would create evolutionary synergy, where the utility of the whole exceeded the sum of its parts. Instead of competing, molecules began to depend on each other to maintain the viability of the protocell as a unit. Such systems over millions of selection cycles could have given rise to the first robust biochemical networks, the protometabolisms, forerunners of modern metabolic cycles. Life, therefore, may not have begun as a fully functional organism, but as an encapsulated molecular community capable of growing, dividing, and evolving, guided by physical and chemical principles that still govern cells today. In the end, protocells represent the moment when matter began organizing around itself. When molecules learned to live together inside a bubble and collaborate to sustain their existence, the boundary between chemistry and biology was crossed. Membranes, these fragile fatty barriers, became the stage upon which life performed its first acts. And although everything started with simple and fleeting structures, they paved the way for the complexity that would follow. Cells with organelles, immune systems, neural networks, and eventually minds capable of looking back and wondering how it all began. The answer may lie in part in those tiny primitive bubbles, once silently floating in Earth's young oceans, carrying within them the secrets of the future. Among the oldest and most intriguing candidates for the title of first form of molecular organization with lifelike behavior are cosevates, microscopic structures formed by aggregates of organic molecules in aqueous suspension. Proposed by Soviet biochemist Alexander Oparin in the 1920s, coacevates are colloidal droplets that form spontaneously when proteins, polysaccharides, or other macromolecules are mixed in aqueous solutions under specific conditions of pH, temperature, and salinity. These droplets are not alive in the modern biological sense, but they exhibit features that bring them close to the concept of life. They have a semi-permeable physical boundary, can concentrate specific molecules inside, respond to environmental stimuli, and in some cases even grow and divide. For this reason, conservates are often regarded as proto-cellular systems, a bridge between prebiotic chemistry and the functional organization of life. What makes conservates especially fascinating is their capacity for spontaneous compartmentalization, which is fundamental for any living system. When molecules like proteins or amino acid polymers interact in solution, they can cluster into denser regions, separating from the rest of the water and creating their own distinct chemical microenvironment. Within these domains, reactions occur more efficiently since reactants are concentrated and shielded from external fluctuations. This means conservates could have functioned as natural microfactories, where fundamental reactions for the origin of life, such as RNA polymerization or peptide synthesis, occurred more frequently and with greater stability. Unlike classic lipid membranes, which require more precise structural organization, conservates arise more simply and under conditions broadly available on early Earth. Another remarkable aspect of conservates is their quasi-biological dynamics. Experiments show they can grow by absorbing material from their surroundings and even divide mechanically into two or more parts, especially when subjected to agitation or temperature changes. Although this process is purely physical, it rudimentarily mimics the cellular cycle of growth and division. Moreover, if molecules with catalytic activity, such as ribozymes or peptides, are incorporated into these droplets, the internal reactions become even more complex and regulated. In certain contexts, conservates even exhibit adaptive behaviors, changing their composition and morphology based on the presence of different molecules in the environment. These traits suggest that they not only could have hosted the precursor reactions of life, but also served as evolutionary platforms where molecular natural selection acted, favoring the most stable and efficient combinations. Conservates also help solve one of the oldest dilemmas of biogenesis: how to keep useful molecules together without them dispersing into the environment. In vast, dynamic oceans, spontaneously formed molecules tend to dilute and react chaotically, hindering the construction of complex systems. By creating spontaneous compartments, conservates concentrate useful molecules, increase reaction rates, and even generate internal chemical gradients that can be used for rudimentary chemical work. In modern laboratories, scientists have already demonstrated conservates that encapsulate functional RNA, fluorescent proteins, and even primitive self-replication systems. This reinforces the hypothesis that simple structures like these may have served as incubators for chemical evolution, hosting the first steps of life in controlled, favorable, and protective environments. Beyond conservates, other types of protocells have been studied as experimental models of pre-life. These include lipid vesicles, protein bubbles, and structures based on synthetic or mineral polymers. Each model has its own advantages and limitations. Vesicles are more stable and resemble modern cells but are less permeable. Conservates are chemically simpler and highly dynamic but lack true membranes. Many scientists suggest that various forms of compartmentalization coexisted on early Earth, each offering specific advantages in different environmental contexts. It's therefore possible that the first living systems did not emerge from a single dominant structure, but from a mosaic of interacting proto-systems, competing and evolving in parallel until one group reached the necessary complexity to become a living cell. The study of conservates and other protocells offers not only a window into the past, but also tools for understanding the very definition of life. They show that life can arise gradually, through smooth transitions between chemical organization and biological functionality. There is no clear line separating the non-living from the living, but rather a continuous gradient of increasing complexity. When molecules began to cluster, protect themselves, replicate, and evolve, even imperfectly, we were already witnessing more than just chemistry. We were witnessing an emergent process of life. And perhaps along volcanic lake shores or in the depths of hydrothermal vents, silent conservates floated, carrying within them the seeds of all that was to come: cells, organisms, ecosystems, and us. Among the frequently underestimated elements in the history of the origin of life are minerals, particularly clays, silicates, pyrite, and other metallic compounds abundantly present in the crust of early Earth. Long regarded merely as passive settings where prebiotic chemistry took place, minerals are now seen as catalytic protagonists in the formation of complex organic molecules. They act as surfaces that guide reactions, stabilize chemical intermediates, organize molecules into patterns, and in many cases accelerate reactions that would otherwise be inefficient or unstable in aqueous solution. These materials not only offer protection against degradation, such as ultraviolet radiation or hydrolysis, but also serve as atomic templates, capable of selecting specific geometries and facilitating complex chemical bonds. In a world without enzymes, minerals may have been Earth's first natural catalysts. One of the most explored ideas is that clays, minerals with layered structures and high ion exchange capacity, may have played a crucial role in the polymerization of nucleotides into RNA chains. Experiments with the mineral montmorillonite have shown that when RNA monomers are placed in contact with this surface, long chains of up to 50 units can spontaneously form. The clay acts as a matrix that aligns nucleotides, allowing them to bond more efficiently while protecting them from degradation by water. This physical alignment, promoted by the clay's structure, primitively simulates the function that today is carried out by highly complex enzymes. In other words, montmorillonite and similar minerals could have been the first natural laboratories on Earth, where critical reactions for the emergence of life occurred with frequency and consistency. Other minerals like pyrite, iron sulfide, are also important in prebiotic chemistry. The iron-sulfur world theory, proposed by Günther Wechtershäuser, argues that surfaces rich in iron and sulfur compounds found in deep-sea hydrothermal vents created ideal conditions for energy-generating chemical reactions, the first metabolic cycles. Pyrite, which forms through the interaction of iron and hydrogen sulfide, releases chemical energy that could have been captured by primitive organic molecules, promoting the synthesis of compounds such as carboxylic acids and amino acids. This means that the origin of life might not have started with genes and replication, but with metabolism first, a set of autocatalytic energy reactions driven by minerals that laid the groundwork for the later emergence of informational molecules like RNA. In this model, life began within the mineral pores of submarine rocks, where natural compartments, abundant chemical energy, and catalytic surfaces converged into an ideal environment. Another remarkable feature of minerals is their ability to adsorb molecules and promote specific spatial interactions, something essential for forming organized structures. Mineral surfaces are neither smooth nor chemically inert. They contain grooves, pores, electrical charges, and functional groups that selectively interact with various organic molecules. For instance, a silicate surface can attract polar compounds and align nucleotides in a way that favors the formation of phosphodiester bonds, the same bonds that make up the backbone of RNA and DNA. Furthermore, minerals rich in magnesium or calcium can act as pH buffers, stabilizing the chemical environment for delicate reactions. In the lab, researchers have observed the formation of more robust lipid vesicles when assembled on clays or metal oxides, suggesting that minerals may also have influenced the formation of the first protocells by reinforcing and shaping their membranes. This multifaceted role of minerals, as organizers, catalysts, and protectors, completely reshapes our view of the early biogenesis scenario. They weren't just a backdrop, but central actors in the chemical drama of life's origin, guiding, selecting, and enabling complexity to emerge. The interaction between organic molecules and mineral surfaces creates a hybrid system that foreshadows modern biology. Rudimentary compartmentalization, specialized catalysis, and spontaneous selection of more stable structures. In environments like volcanic riverbeds, clay mud deposits, or mineral-rich hot springs, these interactions may have taken place over billions of cycles, fostering the rise of autocatalytic systems, proto-molecular codes, and eventually genetic replicators. The philosophical implication of this vision is profound. The Earth itself was the first laboratory, with its rocks, gases, and waters forming a self-organized system capable of producing structures that would evolve into life. The oldest rocks we know carry that silent memory. Each grain of clay, each crystal of pyrite may have witnessed a crucial reaction that led to what we are today. With this, minerals cease to be passive elements of geology and become active chemical agents of a process that had no director or script, but culminated in the most improbable outcome of all: life emerging from stone. In the dark depths of the primordial oceans, where sunlight never reached the seafloor, an environment radically different from Earth's hot surface may have served as the true cradle of life. Hydrothermal vents, also known as black smokers, are geological structures located along mid-ocean ridges where the Earth's crust slowly spreads apart, allowing seawater to penetrate hot rocks from the mantle. This water, heated to hundreds of degrees Celsius and enriched with dissolved minerals such as iron, sulfur, nickel, zinc, and silicon, rises back to the ocean floor and contacts the cold seawater, precipitating minerals that build towering tubular structures. Around these vents, vibrant ecosystems arise, sustained not by photosynthesis, but by chemosynthesis, a process in which bacteria use chemical energy to convert inorganic compounds into organic matter. These extreme environments may have offered the perfect ingredients for the emergence of life. The hydrothermal vent theory, known as the hydrothermal world hypothesis, has gained traction in recent decades, particularly for addressing some of the classical problems of biogenesis. Unlike the Earth's surface, where fragile organic molecules would be exposed to intense ultraviolet radiation and oxidative degradation, the deep ocean floor offers a protected, stable, and chemically active setting. The porous walls of the vents, composed of minerals such as pyrite or greigite, act as natural compartments where molecules can concentrate, react, and evolve. These microscopic cavities would function as proto-mineral membranes, supporting the formation of pH and temperature gradients, both essential elements for metabolism. Moreover, the presence of metallic catalysts and continuous sources of chemical energy, such as hydrogen sulfide and molecular hydrogen, creates ideal conditions for the abiotic synthesis of organic compounds and even rudimentary metabolic cycles. In this scenario, life would not have begun with genes, but with cyclic self-regulating chemical reactions. The hydrothermal world hypothesis aligns with the idea that metabolism preceded genetics. That is, before replicating molecules like RNA existed, there may have been autocatalytic reaction networks capable of sustaining themselves, consuming energy, and generating complex products. Acetate, pyruvate, and other metabolic intermediates could be formed through catalysis on surfaces of metal sulfides like iron and nickel, just as occurs in some modern enzymes, a biochemical coincidence that many view as a direct inheritance from these ancient environments. Instead of conservates floating in shallow pools, life might have arisen as chemistry confined within mineral microcavities, gradually integrating information storage systems like RNA, which would evolve to better control and replicate these metabolic processes. This theory is further supported by the universality of certain biochemical elements. All known life forms rely on metals like iron, copper, molybdenum, and nickel in their most essential enzymes, and these metals are abundantly found in hydrothermal systems. Likewise, many key metabolic reactions, such as nitrogen fixation and ATP synthesis, share features with geochemical processes that occur spontaneously in these environments. Even the structure of ATP, the universal energy currency of the cell, resembles compounds that form under hydrothermal conditions. This suggests that modern biochemistry may be a refined evolution of chemical processes that began in the ocean's depths, a continuity that strengthens the plausibility of life's origin in hydrothermal vents. In fact, some of the most primitive bacteria and archaea known today still live in these extreme environments, feeding on inorganic compounds and thriving without sunlight. However, the hydrothermal hypothesis also faces challenges. One of the main concerns is the stability of RNA and other organic molecules at such high temperatures. While heat facilitates chemical reactions, molecules like RNA degrade quickly under extreme thermal conditions. This has led some researchers to propose a hybrid model based on alkaline hydrothermal vents, which operate at lower temperatures, around 70 to 90 degrees Celsius, and generate gentler mineral compartments such as those found in the vent system known as Lost City. These vents produce stable chemical gradients ideal for molecular synthesis and membrane formation, without destroying newly formed compounds. In this context, the origin of life becomes even more plausible, with molecules being formed, protected, and selected in conditions favorable to the accumulation and evolution of functional molecular systems. The hydrothermal vent hypothesis offers an elegant and coherent vision of life's origin as a continuous geochemical phenomenon deeply tied to planetary dynamics. Earth not only provided the ingredients, but also the geophysical systems that channeled energy and matter into organized structures. More than that, if life can emerge in the dark ocean depths, independent of sunlight and powered only by chemical energy and minerals, it drastically expands the possibility of life elsewhere. Subsurface oceans on moons like Europa and Enceladus, orbiting Jupiter and Saturn respectively, may host similar conditions, making hydrothermal vents not just a terrestrial feature, but a universal template for biogenesis. In this view, life is no longer a rare accident, but a natural consequence of active geology, rich chemistry, and the persistence of deep time. In contrast to theories that place the origin of life in hot dynamic environments, like hydrothermal vents or volcanic pools, a less explored yet deeply intriguing hypothesis suggests that extreme cold may have been the true cradle of life. This perspective is known as the cryogenesis hypothesis, which posits that frozen environments, such as polar lakes, oceanic ice caps, or even volcanic glaciers on early Earth, could have offered unique chemical and physical conditions for the formation and preservation of the first biological molecules. Contrary to common belief, ice is not merely a barrier to life. It also serves as a natural insulator, a concentrator of molecules, and a stabilizer of fragile structures. Rather than being a chemical prison, ice may have been a silent incubator, where time and concentration aligned to allow the improbable to emerge. The experimental basis for this hypothesis lies in the fact that when water freezes, dissolved molecules are excluded from the forming ice crystal and forced to accumulate in thin liquid channels between ice grains, known as eutectic channels. These ultrafine structures concentrate substances by thousands of times, creating highly active chemical microenvironments even at sub-zero temperatures. Furthermore, low temperatures slow down destructive reactions such as RNA hydrolysis, enabling fragile molecules to remain intact for much longer periods. In warm environments, RNA and other biomolecules degrade within minutes or hours. In ice, they can persist for weeks, months, or even years, allowing time for rare and complex processes to occur. This prolonged stability, combined with forced concentration of reactants, turns ice into an unexpectedly efficient chemical reactor for the emergence of life. Experiments with mixtures of nucleotides, amino acids, and lipids have shown that under slow freezing conditions, RNA and peptide chains can form more efficiently than in liquid solutions. The formation of phosphodiester bonds, essential for RNA, is favored in these eutectic microenvironments, where the proximity of monomers increases dramatically. Additionally, the channels between ice crystals can serve as primitive physical compartments, akin to protocells, where specific sets of molecules evolve separately, accumulating distinct characteristics. This allows for the formation of parallel molecular microecosystems where different chemical combinations compete for space, substrates, and energy. Low temperatures also promote the self-organization of lipids into more stable vesicles, forming membranes resistant to thermal agitation and capable of protecting sensitive internal contents. Thus, ice provides not only stability, but also structural architecture for nascent life. Another argument in favor of cryogenesis is the abundance of ice in the universe. Expanding the search for life's origins to other celestial bodies, such as icy moons like Europa, Enceladus, and Ganymede, or exoplanets in cold zones, reveals that ice is far more common than hydrothermal vents or active volcanoes. This raises the possibility that life could emerge convergently in frozen environments, where chemical energy and time are abundant even in the absence of sunlight or intense heat. Comets, asteroids, and frozen moons carry not only water in solid form, but also complex organic compounds like amino acids, alcohols, and hydrocarbons. If cold environments can host complex chemical reactions, then life, or at least its precursors, may be an interstellar phenomenon in potential, repeatedly emerging in conditions that seem inhospitable yet are chemically fertile. Despite its advantages, the ice hypothesis also faces limitations. Reaction rates, though favored in terms of stability, are naturally slower, which means the process may depend on millions of thermal cycles to achieve significant progress. Additionally, energy sources in icy environments are less abundant than in hydrothermal settings. To overcome this, some models propose hybrid environments, such as the edges of volcanic lakes with freeze-thaw cycles or impact craters with surface ice and residual heat. Under these conditions, molecular systems could alternate between preservation in the cold and reactivity in the warmth, creating chemical cycles reminiscent of life's early steps. In this way, cold is not merely a pause, it is a necessary deceleration for complexity to emerge without destroying itself in the process. The cryogenesis hypothesis invites us to rethink our paradigms about what constitutes a life-friendly environment. Instead of warm seas or boiling lakes, perhaps life's first whispers were silent murmurs in icy channels, where molecules met, bonded, and began to dance slowly under the faint light of a distant sun. Ice, so often associated with death and stillness, may have been the most stable geological womb of primordial Earth, allowing the delicate filaments of biology to be born, grow, and evolve, shielded from chaos and destruction. And if this is true, then life is not merely possible in extremes, it may be inevitable wherever time, stability, and chemistry meet in silence. The lipid hypothesis proposes that one of the most fundamental steps in the origin of life was the spontaneous formation of vesicular structures composed of lipids, which may have served as the first containers of life. Unlike theories that place RNA, proteins, or minerals as the main players in the beginning of biology, this hypothesis focuses on the importance of self-organized membranes for the emergence of stable molecular systems capable of growing, dividing, and eventually evolving. Life in all its modern forms is compartmentalized. Whether in unicellular bacteria or complex multicellular organisms, the cell is the basic unit of biological existence, and every cell is surrounded by a lipid membrane. The spontaneous emergence of lipid bubbles, therefore, could have been the turning point between chaotic chemistry and an emerging biological system, marking the beginning of what we now call life. Lipids are amphipathic molecules. They have a hydrophilic end that interacts with water and a hydrophobic end that repels it. When placed in aqueous environments, these molecules tend to self-assemble into spherical bilayers, forming structures known as vesicles or liposomes. This self-organization does not require external energy or genetic instruction. It is governed by basic physical principles of thermodynamic stability. In laboratory conditions, simple lipids such as short-chain fatty acids have been shown to spontaneously form vesicles under a wide range of conditions, including extreme temperatures and pH levels. Moreover, simple prebiotic molecules such as glycerol and acetic acid can give rise to basic lipids in reactions promoted by heat, minerals, or drying and rehydration cycles, suggesting that the ingredients for the first membranes were widely available on early Earth. One of the greatest advantages of lipid structures is their ability to isolate an internal volume from the external environment. This creates microenvironments where chemical reactions occur with more control and less interference. Even rudimentary systems containing just a few catalytic molecules become more efficient when encapsulated, as reactants remain close together and protected from degradation. This kind of primitive compartmentalization not only promotes specific reactions, but also defines a physical boundary, creating space for the emergence of molecular identity, a biochemical self, distinct from the external non-self. Over time, protocells composed of lipid vesicles could absorb nutrients from their surroundings, accumulate useful products, and eventually grow and divide, even without genetic instructions, simply following the physico-chemical rules of diffusion, membrane tension, and osmotic balance. Furthermore, studies have shown that these lipid protocells can encapsulate RNA, peptides, and other complex molecules, shielding them from destructive reactions and increasing their functional half-life. This forms the basis for the hypothesis of a primitive cooperative system in which compartmentalization and molecular catalysis evolved together. In environments with thermal cycles, such as volcanic pools or hydrothermal margins, these vesicles could dry out and rehydrate, promoting fusions, content exchanges, and even spontaneous division. Each cycle would represent a new generation of protocells, with variations in composition and functionality. Those that better preserved their internal molecules, more efficiently absorbed building blocks from the environment, or replicated more robustly, would have selective advantages, even without a genetic code, driven solely by physicochemical selection. An interesting variation of the lipid hypothesis is the possibility that the first living systems emerged from the inside out. Instead of a replicating molecule later finding a protective capsule, perhaps the capsule formed first and eventually encapsulated catalytic systems that happened by chance to favor its own survival. This reversal of logic changes how we think about the origin of the cell. Instead of beginning with information, it would have begun with structure. And that structure would have selected and favored useful reactions until eventually rudimentary forms of molecular inheritance emerged, such as self-replicating RNAs. In this model, life is the consequence of functional containment. Anything that organizes, maintains its shape, and replicates within a physical boundary gains an adaptive advantage, even without intention or planning. The lipid hypothesis also sheds light on a profound philosophical and astrobiological question. If compartmentalization is a universal prerequisite for life, then wherever there is water, lipids, and energy cycles, there is potential for life to emerge. This broadens the search for living or protocellular organisms to worlds with subsurface oceans, such as Enceladus, Europa, or even exoplanets with dense atmospheres. If vesicles can form spontaneously here, they can form anywhere. And once formed, they initiate an almost inevitable process of chemical evolution. After all, anything that is isolated, that grows, and that varies, can evolve. And life, in its most primitive stage, may have begun as a simple fat bubble carrying within it the future of an entire planet. As we reconstruct the possible paths that led to the origin of life, one inevitable question arises with each new discovery. How could something as incredibly complex as a living cell emerge from such simple, disorganized, and chaotic systems? This dilemma is known in scientific literature as the paradox of complexity, and it lies at the heart of the biogenesis debate. On one hand, modern life is astonishingly sophisticated, composed of precise genetic systems, integrated metabolic networks, self-repair mechanisms, and compartmentalized structures. On the other, the prebiotic chemical reactions we know are random, prone to degradation, entropy, and dispersion. The gap between molecular chaos and the functional organization of a living organism seems, at first glance, unbridgeable. But modern science shows that complexity does not need to appear all at once. It can emerge gradually, in successive stages of self-organization, selection, and refinement. One of the key concepts that helps us understand this leap is that of emergent complex systems. In physics, chemistry, and biology, there are countless examples of highly organized structures arising from simple interactions among individual parts. Ice crystals, for instance, form symmetrical geometries from water molecules organizing under physical laws. Flocks of birds create coordinated formations without any leader, merely by following local rules of proximity and velocity. Likewise, in prebiotic chemistry, simple molecules can produce collective behaviors that favor stability, replication, and functionality, all without any plan or guiding intelligence. This property, known as emergence, is essential to understanding how order can arise from apparent chaos. Moreover, biological complexity did not begin with everything we see today. The first living cell was likely much simpler than any modern organism. It lacked DNA, structured proteins, organelles, or complete genomes. It consisted of short RNA molecules, rudimentary lipids, partial metabolic networks, and imperfect replication mechanisms. This protocell would have sustained itself in a fragile manner, making constant errors depending on the environment for its components and undergoing mutations at every division. But paradoxically, this imperfection was the engine of evolution. Systems that replicate with 100% fidelity do not evolve. It is through variation, selection, and competition for resources that complexity grows. Thus, what began as something unstable and nearly random gradually became increasingly robust, precise, and adaptive. Another essential factor in resolving the paradox of complexity is the idea of molecular coevolution. RNA molecules, peptides, and lipids did not evolve separately, but rather in systems where each component influenced the others. An RNA molecule that catalyzed a reaction producing more stable lipids, for instance, favored its own survival. A peptide that protected RNA from degradation increased the chance of replication for that system. These cross-interactions create positive feedback loops, where improvement in one function leads to improvement in another, accelerating the emergence of increasingly integrated structures. In this sense, complexity does not need to be designed. It is naturally selected over millions of years, favoring the most functional combinations, even if they arise initially by chance. The nature of evolution also resolves part of the puzzle. Natural selection is a cumulative process that favors marginal improvements in functionality. There is no need for a final structure to appear all at once. An RNA that replicates imperfectly already has an advantage over one that does not replicate. A lipid that forms stable vesicles is more effective than one that dissolves. A system that maintains a chemical gradient for longer enables more favorable reactions. Over billions of cycles of trial and error, these small advantages accumulate until simple molecular systems evolve into complex ones. Complexity, therefore, is not an inexplicable miracle. It is the inevitable outcome of persistent chemistry under selection. Finally, the paradox of complexity is also addressed by the vast timescales involved. Earth is about 4.5 billion years old, and evidence of life appears within the first 700 million. This means that the evolution of life did not require a single unlikely leap, but rather had sufficient time to explore millions of molecular combinations, continuously tested against the forces of physics, chemistry, and the environment. What we now see as sophisticated biology is just the peak of the mountain. Its base was built slowly, with fragile foundations, but supported by time, variation, and persistence. Complexity did not come from nothing. It emerged, was selected, refined, and eventually became robust enough to leave fossils dominate ecosystems, and finally develop the consciousness to look back and ask, how did it all begin? Among the most provocative theories about the origin of life on Earth is the hypothesis of panspermia, which proposes that life, or at least its precursors, did not originate here but came from outer space. Far from being a fantasy of science fiction, panspermia is a serious scientific hypothesis, supported by astronomical observations, laboratory experiments, and data obtained from meteorites. In its simplest version, it suggests that complex organic molecules, such as amino acids, fatty acids, sugars, or nitrogenous bases, may have formed in extraterrestrial environments like interstellar clouds, comets, or asteroids, and were later delivered to Earth via cosmic impacts. In more daring versions, microbial life itself could have survived space travel and colonized our planet through accidental landings, perhaps aboard rocky meteorites offering protective shielding. The idea of panspermia has ancient roots, but gained momentum in the 20th century with advances in astrobiology and the analysis of carbon-rich meteorites. The most famous example is the Murchison meteorite, which fell in Australia in 1969. When analyzed, it revealed an impressive quantity of natural amino acids, many of which are identical to those found in living organisms. Even more surprising, these compounds showed racemic chirality, that is, they contained both right and left-handed versions of amino acids, a hallmark of abiotic chemical synthesis, yet with patterns that suggest some level of organization. This proves that the formation of life's fundamental building blocks is not exclusive to Earth. Organic molecules can arise in entirely extraterrestrial conditions, whether in cosmic ices irradiated by cosmic rays, in Fischer-Tropsch reactions on early planets, or on sunlit comet surfaces. More recently, space missions such as ESA's Rosetta, which studied comet 67P Churyumov-Gerasimenko, and JAXA's Hayabusa 2, which returned samples from the asteroid Ryugu, found direct evidence of complex organic compounds beyond Earth. Aromatic molecules, hydrocarbons, and even amines, structures linked to biological processes but formed in lifeless environments, were detected. This suggests that the universe is chemically fertile and that life's ingredients may form almost inevitably wherever carbon, hydrogen, nitrogen, oxygen, energy, and time coexist. Additionally, experiments conducted in Earth's orbit have shown that certain bacteria and spores can survive for months or even years in the vacuum of space, especially when shielded by dust or rock. This opens the possibility, still speculative but not impossible, that microbial life could travel between planets, moons, or even star systems, transported by impact ejector or wandering meteorites. Panspermia, therefore, does not directly solve the question of life's origin, but shifts it elsewhere in space-time. Instead of asking, how did life begin on Earth? We begin to wonder, where and when did life first emerge in the universe? In this model, life is not a terrestrial phenomenon but a cosmic one. Perhaps it began on an older planet, a dead star, an ancestral molecular cloud, or even in multiple places simultaneously, like seeds scattered across the cosmos, waiting for the right conditions to bloom. Earth, in that case, was simply fertile soil that received these biological seeds billions of years ago, possibly during the planet's period of heavy meteorite bombardment between 4.1 and 3.8 billion years ago. What we call the origin of terrestrial life might have been merely an episode of biological colonization, either interplanetary or interstellar. It's important to note, however, that panspermia does not replace local chemical hypotheses about life's origin. It complements them. Even if organic molecules or microbial life arrived from elsewhere, they would still have to adapt, replicate, and evolve here, facing Earth's unique environmental conditions. Moreover, life had to emerge somewhere, even if not here. At some point in some corner of the universe, chemistry had to cross the threshold into biology, and that starting point remains unknown. Panspermia therefore expands the scope of inquiry, inviting scientists to look beyond Earth in the search for life's beginnings. And the more we learn about the presence of organic molecules in comets, icy moons, and cosmic clouds, the more the idea of life as a universal phenomenon seems less speculative and more like a logical consequence of cosmic chemistry. Finally, panspermia also stirs our philosophical and existential notions. If life came from space, then we are literally children of the stars, products of a chemical process that began before our planet even existed. This idea connects biology to astrophysics, life to the cosmos, and forces us to recognize that the line between the living and the non-living may be more fluid, broader, and older than we imagined. Life, from this perspective, ceases to be a rare accident and becomes an emergent property of the universe itself, arising wherever matter, energy, and time align. And in that case, the question may not be, are we alone, but rather, how many different forms of life in how many worlds were born from the same cosmic dust that once made us wonder who we are? The evidence that organic molecules essential to life are not exclusive to Earth has been accumulating for decades, and one of the most impressive sources of this cosmic proof lies in meteorites and comets, true messengers from deep space. Contrary to what one might assume, these celestial bodies are not merely sterile rocks drifting through the universe. Many of them carry amino acids, alcohols, hydrocarbons, nucleobases, and even sugars, complex compounds that form the basis of biochemistry. These discoveries support the idea that the chemistry of life is an intrinsic property of matter in the universe, and that natural processes operating on cosmic scales over billions of years are capable of synthesizing the fundamental building blocks of biology without the need for cells, genes, or special planetary conditions. In other words, the chemistry of life predates life as we know it. The most emblematic case of this revelation was the study of the Murchison meteorite, which fell in Australia in 1969. Weighing over 100 kilograms, this carbonaceous meteorite was collected immediately after impact, avoiding environmental contamination. When analyzed, it revealed more than 80 different types of amino acids, many of which are uncommon in terrestrial biology. The isotopic distribution of these compounds confirmed their extraterrestrial origin. Even more impressively, Murchison contained aromatic hydrocarbons, alcohols, esters, and even sugars such as ribose, the same sugar found in RNA. This means that the chemical processes responsible for these molecules were already active in interstellar space, perhaps in cold molecular clouds or on comet surfaces exposed to cosmic radiation. The inevitable conclusion is that organic synthesis is a universal phenomenon and that Earth was not the only laboratory of biogenesis. Comets, in turn, are time capsules that preserve the original matter from the formation of the solar system. Composed of ice, dust, and organic compounds, these structures travel in elliptical orbits for millions of years, interacting with solar and cosmic radiation. The Rosetta mission from the European Space Agency, which studied Comet 67P Churyumov-Gerasimenko up close, detected glycine, an amino acid, ethanol, methylamine, and methyl isocyanate, all compounds associated with the synthesis of proteins and nucleic acids. NASA's Stardust probe, which collected particles from Comet Wild 2, also identified amino acids, reinforcing the idea that the formation of organic molecules in comets is not a rare event. These data indicate that, in Earth's early history, cometary impacts could have delivered tons of complex organic material, serving as essential chemical input for the prebiotic soup. Beyond comets and meteorites, there is growing evidence that complex organic molecules are present in interstellar clouds and protoplanetary disks, such as those observed around young stars. Telescopes like ALMA, Atacama Large Millimeter Submillimeter Array, have detected signatures of methanol, formaldehyde, vinyl cyanide, and other organic compounds in star-forming regions. In some cases, precursors to amino acids and sugars have been identified thousands of light-years from Earth. This means that prebiotic chemistry is not a privilege of our solar system, but a galactic constant. If these compounds are present during the formation of planets and moons, then each new celestial body is born already immersed in organic matter, dramatically increasing the likelihood that, under suitable conditions, rudimentary biochemical systems may emerge independently. The implication of all this is clear. Life on Earth did not start from scratch. Rather than relying solely on the planet's internal chemistry, Earth may have been fertilized by organic compounds from space, delivered by a constant shower of meteorites and comets during a period known as the Late Heavy Bombardment between 4.1 and 3.8 billion years ago. It is estimated that during this time, Earth received millions of impacts, some of which deposited thousands of tons of carbon, nitrogen, and complex molecules into shallow, warm oceans, accelerating the formation of the first replicating molecular systems. This suggests that the origin of life was a hybrid process, combining local ingredients with interstellar material, and that Earth was just one of many places where this cosmic experiment could have taken place. The narrative that emerges from this line of research is profoundly transformative. It invites us to see the universe as a vast chemical reactor, where life is not an isolated miracle, but a recurring possibility. Meteorites and comets are not mere cosmic debris. They are carriers of chemical seeds, natural instruments of universal organic distribution. According to some hypotheses, even Earth's water may have partially come from these objects. Thus, each fall of a primitive meteorite, each fragment analyzed in the lab, each molecular trace detected by a spaceborne spectrometer brings us closer to understanding that life may not be the exception of the cosmos, but its consequence. In the quest to understand how life could have originated under the extreme conditions of early Earth, or even in extraterrestrial environments, one of the most groundbreaking discoveries of modern biology was the identification of extremophiles, organisms that not only survive but thrive in conditions considered lethal to almost all other forms of life. These beings challenge our conventional understanding of biological limits, living in temperatures above 100 degrees Celsius, under crushing pressures, in highly acidic or alkaline pH levels, in extreme salinities, in the presence of lethal radiation, and even without oxygen. Their existence proves that life is not as fragile as once believed, and that the range of habitable environments is much broader than old school books suggested. Studying extremophils is like peering into windows to the past, because many of them preserve biochemical traits that date back to the first billions of years of Earth's history. Among the most well-known extremophils are thermophils and hyperthermophils, found in hydrothermal vents at the ocean floor and in volcanic calderas. They resist temperatures that destroy proteins and nucleic acids in most organisms. To do this, their molecules possess specially stable structures, with reinforced chemical bonds and cell membranes adapted to prevent disintegration. Other striking examples include acidophiles, which live in environments with a pH below 3, such as sulfur mines, and alkaliphiles, which survive in pH levels above 9, like soda lakes. There are also extreme halophiles, capable of growing in salinities higher than seawater, thriving in hypersaline ponds where most life forms instantly dehydrate. All these organisms demonstrate that life is adaptable even at deep biochemical levels, and that their survival mechanisms may have been inherited from very ancient ancestors, perhaps even from the first living organisms on Earth. Even more impressive are radiotolerant and barophilic organisms. The former group includes species like Deinococcus radiodurans, which can withstand doses of ionizing radiation that would kill a human instantly. It repairs its DNA with extraordinary efficiency and has nearly unbeatable antioxidant mechanisms. Barophiles, also called piezophiles, live under pressures above 1,000 atmospheres, such as in abyssal trenches, and possess enzymes that don't denature under intense compression. There are also microorganisms that survive complete desiccation, long-term freezing, and even the vacuum of space. Some of them, like tardigrades, can endure temperatures close to absolute zero and survive in a latent state for decades. This completely reshapes our expectations of what defines a habitable environment and strengthens theories like panspermia by showing that certain organisms could, in theory, survive interplanetary travel encapsulated in meteorites or cosmic dust. But extremophiles are not just biological curiosities. They offer direct evidence about the possible conditions for the origin of life. Many of them belong to the domain Archaea, one of the oldest branches on the tree of life. Their metabolic pathways, use of inorganic compounds as energy sources, and ability to live without oxygen suggest they may be direct descendants of Earth's earliest organisms. They help us understand what a biosphere without sunlight might have looked like, one rich in sulfur, iron, or methane, as existed in the primordial oceans or in the depths of Earth's crust. Their genomes reveal traces of adaptations that may have arisen billions of years ago, allowing us to reconstruct plausible biochemical scenarios for life's origin. More than that, they serve as living models for experimental studies, testing hypotheses about extreme biochemistry, early evolution, and molecular survival strategies. The study of extremophiles also has profound implications for astrobiology. When we ask whether life exists on Mars, Enceladus, Europa, or distant exoplanets, we are no longer limited to seeking Earth-like environments. We now know that life can exist without light, without oxygen, under radiation, in ice, in salt, in acids, or in the dark depths of the ocean. This allows us to imagine alien ecosystems based on alternative biochemical principles, sustained by chemical or geothermal energy sources. If organisms can survive in submerged volcanoes or acidic lakes here, it is reasonable to assume that similar systems could exist on worlds with subsurface oceans, hostile atmospheres, or frozen crusts. Extremophiles, therefore, expand the boundaries of known life and make the existence of extraterrestrial life not only possible, but likely, provided there is stability, organic molecules, and energy. Ultimately, extremophiles teach us that life is not defined by fragility, but by adaptability, that biology does not depend on comfort, but on persistence, that the most resilient organisms are not the largest or most complex, but those that know how to adapt to extreme conditions. By studying these nearly immortal creatures, we touch the deepest roots of biology, and perhaps glimpse reflections of the first living beings that walked, crawled, or floated on this planet. Creatures that not only survived the hellish conditions of young Earth, but transformed it into a fertile garden where billions of years later, conscious eyes would open to gaze at the sky and ask, "Are we alone?" Extremophils suggest perhaps not, and that life, where it can exist, tends to find a way. Long before Earth's atmosphere contained oxygen, the planet was dominated by an anoxic environment, completely devoid of the gas we now consider essential for life. During the first billions of years of Earth's history, the air surrounding the planet was a thick and toxic mix of gases like methane, ammonia, carbon dioxide, water vapor, and hydrogen. Molecular oxygen simply didn't exist in free form because any small amount generated by natural processes was immediately consumed by chemical reactions with iron, sulfur, and other reduced elements in the crust. In this hostile scenario, the first living organisms emerged, creatures that not only didn't depend on oxygen, but wouldn't have even recognized it. In fact, they might have considered it poisonous. Life, therefore, had to develop through alternative metabolic routes, based on other electron donors and acceptors. These ancient anaerobic metabolisms are living relics of a time when oxygen was not only irrelevant, but potentially destructive. Evidence that life flourished in a world without oxygen comes from both geology and microbiology. In the oldest rocks on Earth, dated up to 8 billion years ago, we find traces of fossilized organic matter and carbon isotopes that indicate biological activity in fully reducing environments. And in the current domains of life, there are still obligate anaerobic microorganisms that not only live without oxygen but die in its presence. Many of these creatures are archaea and bacteria that inhabit extreme environments like swamps, marine sediments, animal intestines, and hydrothermal vents. They perform processes such as fermentation, methanogenesis, sulfate respiration, ion reduction, and nitrate respiration, all of which can generate chemical energy without involving oxygen. This demonstrates that aerobic respiration, which uses oxygen, is a later evolutionary acquisition, and that the earliest metabolic pathways were anaerobic, simpler, yet effective enough to sustain entire ecosystems. One of the most primitive and fascinating metabolisms is methanogenesis, carried out by specialized archaea that produce methane as a byproduct. These organisms extract energy by converting carbon dioxide and hydrogen into methane, an exothermic reaction that can occur in moderate or high temperatures in the absence of light and in total lack of oxygen. Methanogenesis is not only viable in many ecological niches of modern Earth, but it also releases methane into the atmosphere, which may have contributed to the early greenhouse effect and helped maintain temperatures above freezing in the planet's early oceans. Another essential process in the anoxic Earth was fermentation, in which organisms break down organic molecules like glucose to obtain energy, generating products like lactic acid, ethanol, or acetate. Simple, self-sustaining and oxygen-independent fermentation is a metabolic legacy still present today in fungi, bacteria, and even in our own muscle cells when oxygen is scarce. In addition, there are respiratory routes based on alternative electron acceptors like nitrate, sulfate, ferric iron, and even arsenic, all of which were abundantly available on early Earth. This form of anaerobic respiration involves electron transport chains that operate without oxygen, yet still create a proton gradient and produce ATP, the universal energy molecule of life. These processes are still observed in bacteria living in mud, underground mines, and deep ocean sediments, and likely resemble the metabolic pathways of the first organisms. This reinforces the idea that life didn't need an oxygenated planet to arise, spread, or evolve. Oxygen, in fact, was a product of life, and its large-scale release around 2.4 billion years ago during the so-called Great Oxidation event triggered a global catastrophe for many of these ancestral anaerobes. This historical turning point shows that the anoxic Earth was the true cradle of biogenesis, a world where the chemistry of life could develop free from the destructive reactions imposed by oxygen. Many of the fundamental mechanisms of biology, such as ATP production, the use of metal cofactors, membrane compartmentalization, and even the genetic code, may have originated in this anaerobic context. When oxygen began to accumulate, it forced a biochemical reinvention that led to the emergence of mitochondria, oxygenic photosynthesis, and aerobic respiration. But the pre-oxidation world was a period of extraordinary evolutionary innovation, where life built its foundations on a planet without air, but rich in chemical opportunities. Understanding this chapter is crucial to recognizing that life does not need ideal conditions to emerge. It only needs stability, energy, and time. Life without oxygen also helps us imagine where else in the universe it might be hiding. Planets and moons with atmospheres rich in CO2 like Venus, or oceans beneath ice crusts like Europa and Enceladus, offer anoxic environments that resemble the Archean Earth. If life emerged on a planet without oxygen, then similar worlds, now considered inhospitable, might actually be alive inside, hosting anaerobic ecosystems that thrive far from light, air, and surface conditions. Earth's past, therefore, is a map for the present of other worlds. And microorganisms that breathe iron, sulfur, and methane may be the best models we have for the kind of life we might one day find beyond our planet. Among the most elegant and deeply geological hypotheses for the origin of life is the so-called iron-sulfur theory, originally proposed by Günther Wächtershäuser in the 1980s. This hypothesis suggests that the first metabolic processes, and perhaps even the earliest rudimentary living systems, did not arise in shallow oceans or surface lakes, but rather deep within Earth's crust, in contact with iron and sulfur minerals, natural catalysts capable of driving complex chemical reactions. In this scenario, life would have emerged not from genes or membranes, but from pre-genetic metabolism, a series of autocatalytic reactions fueled by chemical energy from the planet's interior, long before the appearance of replicating molecules like RNA. Instead of a world dominated by genetic information, this hypothesis proposes an energetic world where chemistry directed organization and complexity, and selection acted upon reaction networks rather than individual molecules. The experimental basis for the iron-sulfur theory lies in the catalytic properties of minerals such as pyrite, Fes2, greigite, Fe3S4, and other metal sulfides found in hydrothermal vents and deep fissures within the crust. When heated fluids under pressure encounter rocks containing iron and sulfur, natural redox reactions occur that release energy that can be harnessed to reduce CO2 into organic compounds. This basic reaction, which transforms carbon dioxide into molecules like acetate, pyruvate, formate, or methane, is analogous to steps in the central metabolism of modern organisms. The implication is fascinating. Life may have begun as a set of chemical reactions rooted in Earth's geology, operating on mineral surfaces that concentrated reactants, stabilized products, and provided a continuous energy source. In this model, the surface of pyrite would be the literal substrate where life was born, the foundational stone of biology. This perspective also resolves one of the greatest dilemmas of biogenesis: how to maintain reaction continuity without membranes, enzymes, or genetic replicators. Instead of cellular compartments, the porous cavities in rocks and mineral surfaces would serve as natural laboratories where reaction cycles took place. Iron and sulfur, in addition to being catalysts, would act as temporary repositories of electrons, functioning as chemical batteries that powered serial reactions. The compounds produced, such as simple organic acids, could accumulate, react with one another, and over time form more complex chains like peptides or ribonucleotides. Natural selection in this environment would not act on organisms or genes, but on the most efficient, stable, and self-renewing reaction networks. Those capable of persisting longer, regenerating components, or producing useful byproducts would be favored, creating a proto-evolutionary system based on mineral catalysis. The iron-sulfur theory also finds support in modern biochemistry. Many of the essential cofactors for life, such as ferredoxins, cytochromes, iron-sulfur clusters, and metallic complexes, have structures and functions directly related to the chemistry of sulfurous minerals. Even nitrogenase, the enzyme responsible for fixing atmospheric nitrogen into ammonia, uses a metallic core of molybdenum and sulfur. This resemblance between biology and geology suggests a direct molecular inheritance, as if living systems had simply internalized mineral catalysis into their own bodies. Rather than inventing from scratch, early life copied Earth's chemistry, turning rocks into proteins, metals into enzymes, and geothermal energy into metabolism. The cell in this model would be a portable crust, a tiny encapsulated fragment of the geological conditions that originally sustained its ancestral biochemistry. From an astrobiological perspective, the iron-sulfur theory has powerful implications. If life can arise from the interaction between heated fluids and metallic minerals in subsurface environments, then sunlight, oxygen-rich atmospheres, or surface liquids are not necessary. All that is needed are rocks, water, internal heat, and the right minerals. This means that icy moons like Europa, Enceladus, or Titan, which possess subsurface oceans in contact with rocky cores, could easily host similar biochemical systems based on geochemical metabolism rather than photosynthesis. In this context, life would be a natural expression of active geology, emerging whenever energetic and mineral conditions align. The existence of entire ecosystems on Earth based on chemosynthesis, such as those surrounding deep-sea hydrothermal vents, is concrete proof that sunlight is not required for life to thrive. The beauty of the iron-sulfur theory lies in its simplicity and coherence. It connects geology, chemistry, and biology in a continuous line, where chemical reactions shape organic structures, which in turn learn to replicate and evolve. In this model, genetic information arises after metabolism, as a way to store catalytic patterns that already worked. RNA and DNA would be chemical forms of memory, capable of preserving and perpetuating the most effective reactions of the living geosphere. Thus, life did not begin with a lightning strike in a puddle, but with millions of years of subtle catalysis on mineral surfaces hidden deep inside the planet, out of sight but close to Earth's warmth. The origin of life, therefore, may have been silent, subterranean, and inevitable, born from planetary dynamics rather than miracles or improbable coincidences. Among the most challenging questions in science is identifying the moment in Earth's history when a collection of disconnected chemical reactions transformed into a cohesive, replicating, and evolving system. In other words, something that not only obeyed the laws of chemistry, but also began to encode, transmit, and perpetuate biological information. This turning point is known as the transition from chemistry to biology, an emergent phenomenon in which molecules begin to carry functional memory, creating self-regulating structures that resemble, even if rudimentarily, living organisms. There was no singular event, no magical spark, no precise instant when life began. What existed was a gradient of increasing complexity, where highly organized chemical systems slowly and progressively began to display the properties we now associate with biology: replication with variation, metabolism coupled with self-maintenance, compartmentalization, and above all, inheritance. This transitional process critically depends on three interrelated elements that form the basis of life as we know it: information, function, and structure. Information refers to the ability to store patterns that can be transmitted. Function relates to the capacity to catalyze or control useful chemical reactions, and structure implies the physical maintenance of a bounded, cohesive system. On the early Earth, these three elements began to overlap in systems such as lipid protocells, containing catalytic RNA fragments capable of replicating or catalyzing their own synthesis. None of these components alone constitutes life. A solitary RNA is merely a polymer. A functionless vesicle is just a bubble. An inert peptide is simply a chain. But when these elements begin to cooperate, exchanging information, catalyzing reactions, and preserving their own integrity, a new biochemical system emerges, poised to become a living cell. At the core of this transition lies the concept of molecular heredity, which becomes possible only when a molecule or molecular network can store a form of functional memory that is passed on, with variations, to subsequent copies. RNA, in this context, emerges as the ideal protagonist. It is both informational and functional. Self-replicating RNA fragments, capable of catalyzing the polymerization of other fragments, form the embryo of what would later become the genome. These molecules need not be perfect, only sufficiently faithful to maintain their function, and sufficiently variable to permit evolution. This establishes the foundation for molecular natural selection, a process where more efficient versions of replicators dominate their environment, initiating a Darwinian trajectory that transforms chemical systems into biochemical populations with distinct lineages, competition, and adaptation. The physical structure of these proto-life forms also evolves alongside their information systems. Lipid vesicles that once formed spontaneously begin to encapsulate more effective replicators, creating protocells with functional internal content. Some of these protocells may have grown faster, absorbed nutrients more efficiently, or divided more regularly, even without sophisticated genetic mechanisms. The competition between protocells with different internal contents and structures paves the way for evolution at the population level, no longer of individual molecules, but of encapsulated biochemical systems. At this stage, chemistry is no longer purely random. It is being shaped by selective pressure, with systems that produce more descendants favored over those that are unstable or ineffective. Life, at this point, is no longer a future possibility. It is a process already underway. Another crucial milestone in the transition from chemistry to biology is the emergence of molecular codes, systems of correspondence between information and function, such as the beginnings of the genetic code. Even before ribosomes existed, certain RNAs and peptides may have established stable relationships between nucleotide sequences and types of amino acids. Experiments have shown that some RNA triplets have direct chemical affinity for specific amino acids, suggesting that the genetic code may have emerged from spontaneous, non-arbitrary interactions. These correspondences could have enabled primitive systems to translate RNA sequences into functional peptide chains, uniting replication, catalysis, and structure in an integrated self-amplifying system. From that point, modern biology begins to take shape. Rudimentary genomes, basic metabolic pathways, growth and division cycles, all still highly imperfect, but functional enough to persist, resist, and evolve. Over time, these systems became more efficient, incorporating proteins as catalysts, inventing DNA as a long-term storage medium, and refining membranes to better interact with the environment. But the fundamental transition had already occurred. Earth, once a world of molecules, now had organisms, still invisible, still vulnerable, but capable of leaving offspring, responding to their surroundings, and chemically transforming the planet itself. The birth of life, in this sense, was not an isolated event, but the accumulation of gradual steps, guided by chemistry, shaped by physics, and solidified by deep time. And although no fossil can capture the exact moment when chemistry became genetics, every living cell carries, in its architecture, the memory of that silent process, when matter ceased to be merely reaction and became replication, information, and inheritance. Long before the formation of the ozone layer, early Earth was fully exposed to a constant bombardment of solar radiation, especially in the ultraviolet, UV range. To many, this radiation might seem purely destructive, degrading fragile organic molecules, inhibiting delicate reactions, and sterilizing exposed surfaces. However, a deeper understanding of prebiotic chemistry reveals that solar radiation, particularly ultraviolet, may have been an essential energy source for synthesizing the fundamental compounds of life. Rather than an obstacle to biogenesis, the young sun and its intense emissions were most likely crucial catalysts for the planet's first chemical reactions. Sunlight, combined with simple atmospheric gases, acted as an activator of complex reactions, generating a rich variety of organic molecules in oceans, ponds, lakes, and shallow atmospheric layers. UV radiation is particularly important because it carries enough energy to break stable chemical bonds, initiating reactions that would otherwise proceed slowly or not at all under ordinary thermal conditions. When it strikes compounds such as methane, ammonia, hydrogen cyanide, or carbon dioxide, all present in the early atmosphere, UV light can generate free radicals, highly reactive molecular fragments that, upon colliding with other molecules, produce complex organic intermediates. Laboratory experiments, including those by Stanley Miller and subsequent researchers, have shown that simulating ultraviolet radiation on gas mixtures analogous to early Earth's atmosphere results in the formation of amino acids, nucleobases, sugars, and lipids. As these products accumulated in shallow water environments or through cycles of wetting and drying, they would have concentrated and been incorporated into more complex molecular systems. Furthermore, ultraviolet radiation can drive primitive photosynthetic reactions, even in the absence of sophisticated biological pigments. Minerals such as titanium dioxide or certain iron compounds, when exposed to sunlight, act as natural photocatalysts, converting inorganic molecules into organic compounds. This light-driven mineral catalysis could have allowed for the conversion of CO2 and H2O into aldehydes, acids, and even nucleotide precursors. In salt-rich regions or clay surfaces, UV light could have activated nucleotides and promoted polymerization reactions, transforming monomers into rudimentary RNA strands. In this way, sunlight not only produced life's building blocks, but also accelerated the construction of replicable structures, bridging the gap between dispersed chemistry and the first genetic systems. It's important to note that the lack of an ozone layer in the early atmosphere, which today shields life from UV radiation, was not an insurmountable barrier, but rather an energetic opportunity. The formation of the ozone layer only occurred after the advent of oxygenic photosynthesis, billions of years after life began. Until then, Earth received intense and constant UV light, and surface environments were immersed in this energy. However, life didn't need to originate in the most exposed areas. Shallow lakes, sediments, air bubbles, shaded coastal zones, or turbid lipid membranes could have acted as natural filters, modulating radiation intensity and allowing only the most useful wavelengths to reach the crucial reactions. This means that the earliest life niches were well positioned to receive just the right dose of solar energy, enough to drive reactions, but not enough to destroy everything created. Solar radiation also played a key role in molecular evolution through chemical selection. Compounds more stable under UV light, such as specific nucleobases and phosphodiester bonds, would have been naturally favored, while less stable ones degraded. This environmental filter may have helped select the biochemical building blocks of life not by chance, but by photonic durability. This could explain, for instance, why RNA and DNA bases possess peculiar resistance to photodegradation, and why phosphate is used as the linkage group between nucleotides, despite the availability of chemical alternatives. In this context, the sun not only provided energy, but also functioned as a chemical selector, shaping the early stages of molecular evolution even before Darwinian gene-based natural selection emerged. Finally, understanding the role of solar radiation in prebiotic synthesis also helps us consider the origin of life on other planets. On exoplanets orbiting active stars such as red dwarfs, or on moons with thin atmospheres, the intensity of UV radiation may be higher or lower than that on early Earth. Assessing how sunlight interacts with atmospheric gases and mineral surfaces in different worlds is crucial for estimating the likelihood of organic synthesis beyond Earth. Thus, studying the role of radiation in the origin of life is not just about revisiting Earth's past. It's about mapping the cosmic boundaries of biogenesis and recognizing that, under the right conditions, light can be both a creator and a guide for life in the universe. As the fields of astrobiology and exoplanetology expand our knowledge of the vastness of the cosmos, one question emerges with increasing scientific urgency. Why, with so many planets, stars, and life's ingredients scattered throughout the universe, have we still found no concrete evidence of life beyond Earth? This enigma, known as the Fermi paradox, highlights the stark contrast between the high statistical probability of life existing on other worlds and the complete absence of signals, contacts, or direct observations. It is what some call the great cosmic silence, a void of responses in a universe teeming with possibilities. In the context of life's origin, this silence raises a profound question. Is life incredibly rare, even with all the necessary ingredients present, or is it common but nearly always invisible, microscopic, or fleeting? One possible explanation lies in the unique complexity of the transition from chemistry to biology. While organic molecules are abundant throughout the cosmos, as evidenced by meteorites, comets, interstellar clouds, and protoplanetary disks, the conversion of these molecules into self-replicating evolving systems may require very specific, stable, and long-lasting environmental conditions. Even if many planets have liquid water and essential elements, factors such as constant geothermal cycles, precise energy balances, suitable mineral compositions, the absence of catastrophic events, and a delicate equilibrium between entropy and organization might be extremely rare. Earth could represent a highly improbable alignment of variables that allowed not only for the emergence of life, but also for its persistence and amplification. If this combination is statistically uncommon, then complex life may truly be an exception, a lone beacon in a sea of unrealized possibilities. Another possibility is that life is, in fact, common in the universe, but exists mostly as silent, hidden, buried, or submerged microorganisms living in subsurface oceans, deep sediments, or hostile environments. If most extraterrestrial life resembles Earth's extremophiles, then we should not expect civilizations, radio signals, or megastructures, but instead discrete biospheres based on chemosynthesis that rarely interact with outer space. This kind of life, though abundant, would leave no detectable marks over large distances and could go unnoticed even during direct exploration by probes or rovers. In that case, life would be a silent, resilient, and local biological phenomenon, more akin to bacteria than humans, and Earth an exceptional communicator in a universe of cellular silences. There's also the hypothesis that life is common, but technological intelligence is rare. Earth's own history illustrates this well. For nearly 4 billion years, the planet was inhabited solely by unicellular organisms. All the complexity of modern civilization arose in the final seconds of the cosmic clock, resulting from a series of unlikely evolutionary events, such as the emergence of the eukaryotic cell, multicellularity, symbolic cognition, and language. Each evolutionary milestone required mutations, selective pressures, and environmental stability over hundreds of millions of years. If even one link in this chain had failed, Earth would still be a world of microorganisms. This suggests that simple life may be common, but intelligent life is a statistical rarity, and that's why we have neither heard nor seen its signs. Cosmic silence may also be explained by cultural, temporal, or technological factors. Advanced civilizations may choose not to transmit signals out of fear, ethics, or pragmatism. Others might use technologies incomprehensible or undetectable by our instruments. There's also the issue of timing. The window in which a civilization emits detectable signals could be extremely brief, especially if it self-destructs through war, climate change, or technological collapse. In this scenario, the universe would be a cemetery of extinct civilizations where the time between birth and silence is too short for us to overlap with any of them. Silence, then, would not reflect a lack of life, but a lack of synchronization between the seekers and those who once existed. Finally, there is the unsettling possibility that Earth is truly unique. That, for some reason not yet understood, be it statistical, physical, or even metaphysical, we are the only point in the cosmos where matter awakened into consciousness. This idea, though controversial, cannot be entirely dismissed, since thus far, all empirical evidence points to Earth's biological solitude. But even if that's the case, the very existence of life here transforms the entire universe. In that context, we are the means by which the cosmos observes itself, a rare spark of organization capable of contemplating its own origin. And this confers upon us a singular responsibility to protect, study, and understand life as the most precious and rare phenomenon ever detected, perhaps the only one. After billions of years during which life was limited to simple unicellular forms, something extraordinary occurred in Earth's primitive oceans, a radical acceleration of biological complexity, marked by profound cellular innovations, unexpected symbioses, and genomic reorganizations that forever changed the course of evolution. This leap, known as the transition from unicellular to complex multicellular life, was not rapid, but it was decisive. The foundation of this advancement lay in the invention of the eukaryotic cell, a highly organized structure with internal compartmentalization, specialized organelles, and, most importantly, the presence of mitochondria. Without this crucial step, which involved a symbiosis between distinct organisms, life would never have moved beyond the boundaries of microscopy. It was from this innovation that the paths opened to the diversity of plants, fungi, animals, and eventually, humans. The eukaryotic cell is not just a larger cell, it represents a completely new biochemical architecture. Unlike bacteria and archaea, which have simple internal structures, eukaryotes possess a binucleus enclosed by a membrane, a dynamic cytoskeleton, organelles like the endoplasmic reticulum, Golgi complex, lysosomes, and above all, mitochondria, which are in fact domesticated symbiotic bacteria capable of generating energy with remarkable efficiency. This symbiotic relationship, known as the endosymbiotic theory, posits that an ancestral cell engulfed an aerobic bacterium, but instead of digesting it, began to cooperate with it. The result was our invention of a cellular energy factory, enabling cells to grow larger, regulate their interiors, and coordinate metabolic activities with unprecedented precision. With more energy available, the cell could accommodate more extensive genetic information, more complex regulation, and structural innovation. paving the way for life forms never before seen. With more sophisticated cells, the possibility of unassociation between individuals quas emerged. Multicellularity began simply, colonies of cells that did not fully separate after division. But over time, these colonies began to specialize their cells, creating rudimentary tissues, functional gradients, and patterns of biochemical communication. The key to this transition was stable genetic cooperation, where all cells shared the same genome and did not compete individually. This allowed for the emergence of organisms like multicellular algae, sponges, and eventually animals. This shift from the individual to the collective marked the beginning of an explosion of evolutionary diversity, culminating in the famous Cambrian explosion, when nearly all major animal phyla appeared within a relatively short geological interval, heralding the origin of brains, eyes, exoskeletons, and complex digestive systems. But the transition was not only structural, it was genetic and regulatory. The increase in the number of genes alone does not explain the complexity of multicellular organisms. What truly changed was the way genes were activated, combined, and silenced. Gene regulatory networks, based on transcription factors, small RNAs, alternative splicing, and biochemical feedback, allowed a single genome to produce hundreds of different cell types. This regulatory sophistication was crucial for embryo formation, cellular differentiation, coordinated growth, and tissue repair. The cell, once an autonomous unit, now became part of a collective organism, obeying chemical and genetic signals, communicating via hormones, neurotransmitters, and direct contact. The multicellular organism began to function as an integrated entity, capable of responding to its environment with increasingly refined behavioral and physiological strategies. The emergence of complex life also had geological and planetary consequences. Multicellular organisms altered biogeochemical cycles, modified the atmosphere, promoted massive-scale carbon and nitrogen fixation, and introduced new vectors of erosion, sedimentation, and oxygenation. Biodiversity increased ecosystem resilience, drove coevolution between predators and prey, and established the first stable ecological systems. This was possible because the eukaryotic cell enabled host organisms with longevity, sexual reproduction, and adaptive memory, exponentially expanding the evolutionary potential of life on Earth. What had once been a planet of silent, discrete microorganisms became a vibrant tapestry of forms, colors, strategies, and behaviors. Biology had surpassed the molecular scale. It now operated on the level of the visible, the dynamic, and the interconnected. The history of complex life is, therefore, a story of cooperation, integration, and energetic innovation. Every eukaryotic cell carries within it the memory of that original symbiotic fusion, and every multicellular organism is a testament to the evolutionary power of internal collaboration. By understanding how the cell became an organism, and how organisms became ecosystems, we recognize that life is not merely an efficient chemical reaction, but an emergent process of hierarchical organization, collective adaptation, and the long-term construction of complexity. And all of this began with the humble ability to generate more energy and use it to maintain order, an ability that changed Earth forever. If we consider the vastness of cosmic time and the relative youth of human intelligence, we are compelled to contemplate an intriguing possibility. What if we are only at the beginning of life's story, not its climax? Life, as a universal phenomenon, may very well be just taking its first evolutionary steps. Earth is 4.5 billion years old, and humans have existed for a mere 300,000 years. The entirety of civilization fits into a fraction of a second on the geological clock. This suggests that, despite all the complexity we've developed, we may still be just a primitive form of intelligence, a temporary prototype of something that, with time and life's persistence, will evolve into levels of consciousness, organization, and perception still inconceivable. In this view, life is not the culmination of creation, it's the starting point of something far greater. Biology itself offers precedents for this idea. For nearly 5 billion years, life on Earth was dominated exclusively by microorganisms. Multicellular complexity, brains, eyes, social behaviors, and language are all extremely recent and rare innovations. This indicates that nature is in no hurry and that deep evolutionary processes operate on timescales far beyond our emotional comprehension. It is possible that intelligence, as we currently know it, is still in an early phase, marked by instability, impulsiveness, and self-destruction. Technological civilizations, upon emerging, may go through turbulent transitional phases, like ours, periods in which the power to alter the planet exceeds the wisdom to preserve it. If we survive this phase, we might become something more enduring, more harmonious, more conscious, and less human in the current sense of the word. This hypothesis raises a crucial question. Does evolution have a direction? The most accepted answer in science is that evolution has no intrinsic purpose, but rather a tendency toward complexity in certain contexts, especially when available energy allows for more efficient organized structures. If this tendency continues, and if life overcomes its existential bottlenecks, such as wars, environmental destruction, and cultural collapse, it is possible that we will witness hybrid forms of life, symbiotic unions of biology and technology, distributed intelligence, expanded consciousness, planetary or even interstellar neural networks. The emergence of artificial intelligence, neural interfaces, and biotechnology suggests that evolution is no longer driven solely by mutation and selection, but also by engineering, culture, and collective learning. This acceleration may usher in a new age of life, the post-biological era, where the boundaries between living and non-living matter, between brain and machine, between individual and network, blur into indistinction. And if we are at the beginning, then biogenesis, so mysterious and debated, is merely the foundation of a long cosmic staircase, with each step representing an expansion of what life can become. Prebiotic chemistry led to RNA. RNA led to cells. Cells led to organisms. Organisms led to minds. Minds led to culture. And now, culture is beginning to create technology capable of rewriting biology itself. We are ascending an evolutionary ladder that may lead to levels of complexity and harmony, capable of shaping not only planets, but solar systems, galaxies, and perhaps, one day, the very fabric of the universe. Every breathing cell, every firing synapse, every emerging idea may be a seed of a future we can barely imagine. That's why, when we study the origin of life, we are not merely looking into the past. We are observing ourselves as a phenomenon in motion, still unfinished, still fragile, but with extraordinary potential for transformation. The question is no longer just, how did life begin, but rather, what can life still become? And perhaps right now, as we contemplate this journey that began with molecules at the bottom of the ocean, we are taking the first conscious steps towards something Earth has never seen, and the universe awaits in silence.